All right, I am delighted to welcome everyone to the next installment of our Science of Childhood Cancer lecture series. And I'm really truly thrilled to have the honor of introducing Dr. Nada Jubato as our speaker today. Dr. Jubato is a professor of pediatrics at McGill University and a pediatric neuro-oncologist at Montreal Children's Hospital. She completed her residency in pediatrics with a specialization in hemato-oncology and also obtained a PhD in immunology in Paris. She followed that then with a postdoctoral fellowship in biochemistry at McGill and began her career as an independent investigator at McGill in 2003, where she's pioneering a world-class research program in pediatric brain tumors and has made, as you'll see, a number of seminal discoveries. Dr. Jabato's groundbreaking work, part of it in parallel initially with Susie Baker's lab at St. Jude, has really created a paradigm shift in cancer with the identification of histone mutations in human disease, which has contributed to a transformation of the understanding of the mechanisms by which chromatin alterations can drive cancer. And Dr. Jubato has followed this up with just fantastic stories. She's authored over 220 peer-reviewed publications, many of them in top journals, and she has over 33,000 citations and an H-index of 91, very high. Dr. Jubato has been inducted as a Fellow of the Royal Society of Canada, is an appointed member of the CIHR Governing Council, and holds a Canada Research Chair in Pediatric Oncology. She's clearly an international leader in the field of neuro-oncology and cancer, and I'm delighted to host her today in the Science of Childhood Cancer. Just a couple of housekeeping notes before we begin. If you have any questions at any time during the talk, just type them in the chat box and send them to all panelists. I will keep track of them and then be able to share them with Dr. Jubato as we end. She's also graciously agreed to stay on past the top of the hour. We'll announce the top of the hour, but if there are additional questions after that time, she will be happy to answer them. And so with that, I will turn it to you, Nada.
Thank you very much, Charlie, for the very kind introduction and for the opportunity. It's a little bit kind of challenging to speak to your computer, but at the same time, I think this is a good way also to these days to convey information and to kind of interact with your peers. You know, when you speak about what I've done, this is something that people at St. Jude, I share a lot of things with Susie, with Steve Mack, with Paul Northcott, with you, Charlie, you with your work on the Swift Sniff is quite amazing, and really, it's our passion to kind of understand better what kind of drives those deadly tumor that we share that's brought me here today. So without further ado, today what I'm going to try and tell you is that I thought that, you know, we were in a long, long dark tunnel. At last I'm seeing, and this is my disclosure, I have no financial disclosure, but I start seeing a light at the end of the tunnel, because our understanding, the work that has led to the discovery of the histone mutation that I'm going to be speaking to you about and how to treat them, we're almost there. We're almost there, and I hope that it's going to go very fast as of now. So the histone mutation, the oncohistones, as Dave Alice coined this term, is a discovery I share with Susie Baker's group. And in 2012, we identified it in those high-grade gliomas. And now there is more than that. And what I'm going to be kind of sharing with you is a little bit what we know. There is more than those 3 residue on this histone 3 tails that have been uncovered as being mutated. But for today's talk and for today's sake, I'm going to focus on those 3 residues, and probably more specifically on one of them, the K27. And based on the time, I'll speak a little bit more of the others. So lysine 27 that changes to methionine and more rarely to an isoleucine on any of the histone 3 variant that's found as a somatic mutation in high-grade brain tumors. A glycine 34 that changes to an arginine or a valine in the brain, and believe it or not, never in the brain to a tryptophan or a leucine elsewhere in some bone tumors. And a lysine, and this glycine 34 mutation is exquisitely and exclusively associated with the non-canonical histone 3.3. And the lysine 36 also changed to methionine, bizarrely. 27 to methionine, 36 to methionine on the lysine, also on any of your histone 3 variant. And those are the 3 oncohistones I'm going to be speaking to you about today. And we adopted a new oncohistone mimic. And EZIP, EZH interacting protein, was uncovered as highly expressed or mutated by David Allison from St. Jude and Marcel Kuhl. Her beautiful work in Acta neuropathologica in posterior fossa group A ependymoma, a tumor that has no recurrent alteration except for this expression, aberrant expression of EZIP. And a work in which we collaborated from Peter Lewis's lab, we showed that this EZIP has actually a 15 amino acid sequence that's conserved throughout any species that expresses placental mammal that express EZIP, that mimics actually a lysine 27M on a histone 3 tail, which leads us to say that EZIP is actually a K27M mimic, or maybe K27M is an EZIP mimic. So there's one thing that I want to enrich. So those, where were they discovered, those oncohistones? There is high specificity to the cancer, and within a cancer, to the specific location those mutations occur in. What do I mean by that? The lysine 27M changes on any of your histone 3 tail and EZIP are exquisitely seen mainly in the brain midline, and specify, I hope you see my arrow, the diffuse midline glioma. The pons, histone 3.1, K27M, and histone 3.3, K27M. The thalamus and the spine, histone 3.3, K27M. And when you don't find any of those K27M on histone 3 tail, you find EZIP abnormal expression. And in the temporoparietal cortex, a little bit sometimes in the frontal, you have the glycine 34 mutation to an arginine or a valine on histone 3.3. You never find those glycine mutation in the midline. And even if there's been 1 or 2%, I presume those are probably metastasis from a midline tumor, you never find K27M in the cortex. And I think this means something. Similarly, the EZIP expression, aberrant expression in posterior fossa group A ependymoma is exquisite to the posterior fossa. This is where it's really right next to the brainstem where those ependymoma, posterior fossa group A ependymoma arise. And when you don't find EZIP, you find K27M mutation. And those two never coexist together, which leads us to say that they are actually honing in to do the same effect. We find K27M mutation, and this work was done by the group of Guy Sauvageau, and we also identified it in acute myeloblastic leukemia. And actually, 7% of myelodysplastic syndrome have K27M mutations. And EZIP was also aberrantly expressed, and this was identified by the group of Ali Latifar in endometrial cancer. And those are mainly fusions that lead to aberrant expression of this gene that really is superbly restricted in where we don't even know when it's normally expressed except for the germ cells. And germinoma, germ cell tumors have high expression of EZIP. The G34 mutation that are found in the bone, and this work was done by the group of Adrian Flanagan and Peter Gamble from Oxford, are in the giant cell tumor of the bone, where 92% of those tumors have either a tryptophan and very rarely leucine, exquisitely in histone 3.3. And they also identified in 96% of chondroblastoma, which is another benign tumors of the bone, a lysine 36M. And this was the first identification of a lysine 36M mutation. And we, in collaboration with a group of David Alice or with Jacek Majewski, identified it in head and neck cancer, in the oral cavity, actually, of head and neck cancer that are HPV negative, and in 20% of undifferentiated sarcoma. And those are the main tumors where you find any of those oncohistone being expressed. There will be others, but we've been kind of seeding the literature as it's coming. And for now, I can say with certitude that those are the tumor where we identify those histone mutation to occur. So how do they promote cancer? First of all, just a few words, and I won't bore you more with that. The lysine 27 is a very important mark on a histone 3 tail. It gets monodi and trimethylated by the PRC2 complex, and EZH1 and 2 are the metal transferases that mediate this. And when it doesn't get methylated, it gets acetylated, and lysine 27 acetylation is really a beacon for active transcription and to start actually transcription and recruitment of the polymerases. The glycine 34 has zero post-translational modification, but your glycine, your smallest amino acid, and a lot of work has shown that the change to bulky amino acid probably interfere with the position or the reading of a post-translational modification on lysine 36. And lysine 36 gets mono, di, and trimethylated by multiple different enzymes, the NSD1 or 2, or by SETD2 mutation, which is the only lysine 36 trimethyl transferase. And we identified truncating mutation in SETD2. I apologize for the noise, in hemispheric high-grade gliomas. And what does it do? When a K27 is trimethylated or dimethylated, and it gets deposited on your euchromatin. And this is a repressive mark. The trimethylation of lysine 27 will recruit the PRC1 complex and will induce compaction and silencing of genes when it is on a promoter, but it also induces compaction in the intergenic space. The lysine 36 is associated more with active transcription. The trimethylation goes into gene bodies and associated with active transcription, and the dimethylation is also an active mark, or presumably an active mark. And there's something that's quite key to understand from the beginning, is that those marks, the lysine 36 when it's methylated and the higher level of methylation, and the lysine 27 when it is methylated and the higher level of methylation, they do not coexist together. So they are really putting specific boundaries on your chromatin, where one mark, this is my territory, you don't come into it. And actually the lysine 27 trimethylation mark is a very pervasive and invasive mark. And once it's deposited, it's impossible to deposit the lysine 36 di. And people now are viewing the lysine 36 dimethylation as a way to prevent or to slow down the spread of the lysine 27 trimethylation. And believe it or not, those are the two marks that are absolutely wrong due to those oncohistones. The other level that I want to bring, because this is what I'm going to be speaking about a little bit later on, and some new work that just got accepted, is how come you have in histone, you have any histone 3 that can get a lysine to a methionine modification, and exquisitely histone 3.3 for glycine 34. And the other story is, does it matter if K27M occurs on histone 3.1 or histone 3.2 or histone 3.3? And the reason why I say that is that the pattern of deposition on the pattern of production of those histone 3 variants is completely distinct. Your histone 3.3 is non-canonical. It's present throughout your cell cycle, doesn't need the cell to divide in order to be there and to be deposited. Whereas the histone 3.1 and 2 are canonical and need the S phase to be produced and to be deposited, and they go mainly on your chromatin. Whereas the histone 3.3 is the major histone 3 variant that gets deposited on pericentric heterochromatin, where you have a lot of ERVs, for example, and on your telomeres. And there are specific complexes that mediate the deposition of histone 3.3 in these areas, ATRX DAX, versus euchromatin, which is here a complex. Start to be complicated, but I promise I'll render it more simple. So there are so many questions. How K2M promote oncogenicity? Is there a difference between the different G34 variants? Are there differences between ESIP, histone 3.1, histone 3.3, K27M? Why the cancer specificity? And what I haven't told you, there is a very specific spatial-temporal window specific permissive for oncogenicity. And other question that I won't have the time to go over, but that the discovery of those oncohistone provided. Let's start with the K2M substitution and ESIP and how do they promote cancer. The first work, and I have to say it, the beautiful work that was done by Peter Lewis when he was a postdoc in Dave Alice's lab, opened the way to our understanding on how those K27M or K36M mutation were occurring. And before even the K36M were uncovered, what he showed is when you change a lysine to methionine on any of your residues of the histone 3 tail, what happens is the product inhibit the respective metal transferase that deposit the mark. So your lysine 4 cannot get properly methylated. Your lysine 9 cannot get properly methylated to lysine 27 and the lysine 36 by extension, because what you create is almost like a glue that makes the metal transferase domain very not easily accessible, and your metal transferase is very, very slow at depositing those marks and the higher level of these marks. So we collaborated with a group of Dave Alice and Peter Lewis, and I start with the K36M. Why? Because this mutation was identified on its own without any association. And when we started working, and we had discovered this mutation in 2011, and really couldn't get a sense of what it was doing, the K27M. So we started with K36M because it seems to be on its own and potentially easier to understand. And Xiaolu, who was a postdoc in Dave Alice's lab, led this work. And what he did, he overexpressed the K36M in mouse mesenchymal stem cells. And any of these expressions, whether it's the K36M or the K36I, led actually to a blockade of differentiation. When you push those mesenchymal stem cells to differentiate into adipocyte, chondrocytes, or osteoblasts, the differentiation could not happen. And that was specific to K36M because other substitution to NA or to arginine didn't do the same trick. And when we looked at the chromatin, this is the first hint of news of how this is happening that were provided. This is 3 chips, and this is just representative, and it's true genome-wide. Lysine 36 trimethylation in a wild type gets deposited in the gene body. You could see those beautiful here. The lysine 36 dye is a very abundant mark in the intergenome, and the K27 tri doesn't get deposited where you have K36 dye. As you could see, I hope you could see my arrow, and you could see that they are mutually exclusive and they specify specific boundary. You introduce the K36M as expected, you really decrease drastically the level of lysine 36 tri and lysine 36 dye. But look what happened to the K27 tri. The K27 tri loses its boundary. The thing that was preventing its spread is gone. So this mark goes and spreads within the intergenome. But remember, everything is in rate limiting quantities. The PRC2 complex is not present in more abundance, it's the same. And depositing the mark, because it has way more area, becomes a little bit less effective. What do I mean by that? So even though you're depositing the K27 tri in the area where it should be very highly deposited, very richly deposited, you have a decrease, a relative decrease in the deposition. And why is that important? Then you have lesser. This is bulk. You're not looking at the single cell. And you have a decrease, a net decrease in the recruitment of the PRC1, which is needed to silence and compact your chromatin and silence those genes. So actually, what you're doing is you're diluting ring bond B the same way because it has other area and it doesn't get recruited to the areas where it should go. And I'll show you how. So you have a genic region that is lighting up with K27 tri and intergenic region that also have a lot of K27 tri, recruit your polycomplex. Your gene here is off because it's compacted, and here it's a compacted area, and the boundary with the lysine 36 dye that's deposited by those NSDs is here. In the context of K36M, you're losing the dimethylation in the intergenome. You're increasing the number of K27 trimethylation in there. And by extension, there are 2 things that are happening. The level of tri may go lower, but even if they weren't going lower, your PRC1 has so much more targets that it gets spread, it gets diluted, and gets hijacked. So this gene that should be silenced, and it may be a stemness gene or an oncogene, gets expressed. And those areas that should be expressed or active for enhance or other things gets unduly repressed. And this is what promote actually sarcoma development probably, and those chondroblastoma that have K36M mutation. Okay, we're interested in K27M. What happens with K27M? If you look at an immunohistochemistry of a K27M mutant DIPG here in this case, you have the wrong impression that you have no K27 trimethylation being deposited. It's completely devoid. Here is a wild type sample with the brown shows the K27 trimethylation. And here the rare brown nuclei are actually either immune cells or blood vessels that are wild type for the mutation. Okay, this is a wrong impression because actually what happens, and this is the first cue as to the fact that those K27M mutants are not real EZH inhibitors. There is still deposition of K27 tri, and to some level, deposition and spread of K27 di. So there are still areas that have K27ME3. They were called islands. A lot of speculation came from this residual deposition of K27 tri, and I won't go into that for the sake of time. Just to say that EZIP, it's exactly the same. And what happens is that when you want to deposit the mark, you recruit your PRC2 to specific PRC2 target, and those are mainly CPG island, because this is the highest affinity for those polycomb complexes. And you start depositing the mono, the di, and the tri, and the more level of methylation you have, the di and the tri, this is way more exciting for the enzyme, needs more time. So it gets deposited as much as it can within a given cell cycle, and it's spread from the area of the nucleation. So you have the hit of your PRC2 that start depositing those marks, and then it runs from those areas, those nucleation areas. What we showed, and this is something that was also shown by Peter Lewis, and we collaborated in that for EZIP, is that your PRC2 in the context of K27M or in EZIP is able to hit your chromatin in the area where it should hit it in the cell of origin or lineage of origin. But this enzyme for the trimethylation and to some level for the dimethylation cannot run as effectively. So you are really restricting the di and mostly the trimethylation to K27, to those PRC2 nucleation sites. This data, all of this data was acquired on histone 3.3 K27M. And a lot of speculation came as to why, whether EZIP is different, histone 3.1 and 2 are different, and if there was any variability with the anatomical location. And this is work that was just accepted in Nature Genetic that I'm sharing with you, and it was co-led by Céline Jessa, a very talented graduate student in Claudia Kleinman's lab, Ashat, who was a previous postdoc in my lab, and Shakur, a graduate student was in my lab, and Maud Hillswit, also a graduate student in my lab. So we asked ourselves the question, okay, we find histone 3.1 K27M exquisitely and exclusively in the ponds, and a lot of data, not only from us, but from many others, including Susie, Chris Jones, Michel Monge, and we name them. Those are in younger kids, and they are exclusive to the ponds. The histone 3.3 K27M is present in the ponds in a little bit of older children, but it's also present in the thalamus. And in the thalamus, you could even have adults up to the age of 50 that have K27M tumors. And you find it also in the spine and in the cerebellum. Are they actually specifying distinct progenitors? Are they tumors that migrated from one site? Is histone 3.1 different than histone 3.3? And by extension, is EZIP, the rare tumor that have EZIP, and EZIP in posterior fossa ependymoma, different than there? And so we tried to tackle this. How did we do it? And why did we do it? If you look at the K27M histone 3.3, the group of Steve Henikoff showed on one cell line, that histone 3.1, that there was barely any K27 tri or K27 di deposited in the context of the histone 3.1 K27M mutation, and their conclusion is that it was really way more severe than histone 3.3, and it was shaping differently the epigenome. The group of Michel Monge, also using another approach, concluded that potentially there is a different enhancer oncogenic landscape associated with histone 3.1. And our work, and it's mainly Peter Lewis's work, we only collaborated in that, he showed that actually EZIP is even more strong in vitro in inhibiting PRC2. So what we did, we took cell lines and tumors that came from patients that were either the beautiful yellow that you see here is histone 3.1 K27M. The red is histone 3.3 K27M, and the purple is posterior fossa ependymoma that have aberrant expression of EZIP, except for one sample, which is called PS113004, that is a K27M histone 3.1 ependymoma. And when you look at the deposition of K27 tri, you look at the level, you see that despite the fact that presumably EZIP is stronger in inhibiting in vitro PRC2, the level of K27 tri are higher. And the same goes for K27 di. And histone 3.1 K27M have the lowest level of trimethylation by mass spec. And this work was done in collaboration with Ben Garcia's lab. And when we looked at the pattern of the position, it's very peaky. Exactly the same thing, restricted to the CPG island, to what we think is a nucleation site. Have a look at what happens with the K27 dye. If you look at the K27 dye, the posterior fossa group A ependymoma have close to normal. Those are really similar to what you see in the posterior fossa group B ependymoma or supratentorial ependymoma, the normal spread of the K27 dye. And you see that the spread of the K27 dye is a little bit more in the histone 3.3 compared to the histone 3.1. And the only sample, and I hope you see my arrow, that is a histone 3.1 K27M behaved like an ependymoma, which tells me that this deposition of the dye and potentially the tri is not due to the mutation, but what's happening in the lineage or the cell of origin. And one of the ideas, because as I'm telling you, the higher the methylation level, the more time your PRC2 needs to stay in order to deposit properly the trimethylation. And we even evolved to have a speed up mechanism which in the form of EED, which allows for allosteric activation of PRC2 and the spread of the K27 trimark. Beautiful work done by the group of Danny Riedberg and Raphaël Margeron, who led this work on EED. So the bottom line is that you need more time to deposit the trimethylation. And so we asked ourselves the question, is it because those cells have different cycling rate and potentially different boundaries that would actually prevent them from effectively depositing and spreading the dye, because the tri is similar in its pattern? And so what we did the following experiment. We took a histone 3.3 cell line, that the K27M mutant histone 3.3. We CRISPRed the mutation, and then we overexpressed the histone 3.1. If the histone 3.1 K27M is more drastic, it should actually have us have a dimethylation that's very restricted and decrease in the level of the trimethylation. And new sites, because people were saying that histone 3.1 K27M, because it's histone 3.1 are deposited elsewhere than histone 3.3, that, this is where the oncogenic enhancer landscape was arising. We saw zero difference in the location of the trimethylation mark of K27. And the same thing in histone 3.1. And the beauty is for the histone 3.2, it was exactly deposited than as if it was a histone 3.3. Not more restricted, potentially even a little bit more spread. And what we also did the reverse. We took a cell line that was histone 3.1 K27M and had an ACVR1 mutation. ACVR1 is a growth factor that accelerates the cell time proliferation. And when we removed by CRISPR the ACVR1 mutation, what happened is that those cells proliferate really very much slower. And we looked at the K27 trimethylation. We could, even if it was slower, we had close to similar level of K27 trimethylation and not new sites being deposited. But the dimethylation was impressively, completely increased, which lead us to say that what those mutations do is they hone in on restraining the spread of K27 tri. And the K27 dimethylation, its spread is actually a function of what was the boundary in that cell. And I have more data to show for the boundary, but just for the sake of time, you need to believe me on my word. And this is also work that was done by the group of Danny Reinberg in a beautiful paper in Science Advances 2 years ago, where he showed that the dimethylation is really one boundary that is important. So bottom line, you restrain K27 tri, and the dye goes as far as your cell cycle or your boundary would allow it to do. And the why are we summing this? What happened to the boundary? And are there differences in the cell lineage of origin based on the tumor location? Now that we know that those mutations are doing the same thing, which is converging on restraining K27 tri to the nucleation site, is there differences in cell of origin? So first question, easy question. What replaces intergenic K27 tri and dye in those cells? The first clue came from work that I co-led with Steve Mack, who now is with you at St. Jude, and Claudia Kleinman, where we showed that the dimethylation was actually replaced in the context of histone 3.3 K27M by pervasive acetylation. So what happens, and it's not our work, the dimethylation has been shown to be an important mark that coats your chromatin, and it prevents the undue recruitment of histone acetyl transferases. And the loss of this lysine 27 diene area allowed those histone acetyl transferases to deposit this pervasive, what we call pervasive acetylation in the intergenome, and have a baseline of activation of endogenous retroviruses that would have been otherwise silenced. And that you could potentially push them a little bit further by removing other layers that restrain the expression of those endogenous retroviruses like DNA methylation, for example, or increasing the level of acetylation by using HDAC histone deacetylase inhibitors, and promoting viral mimicry and making the cell look hot to the immune system. And work that we have in our paper now shows that either posterior fossa group A ependymoma do not have pervasive acetylation. Why? Because their coating in lysine 27 dye is completely intact. So there is no room for hats to be recruited. And the histone 3.1 are quite able to recruit it, except when they have lysine 36 dye being highly expressed. And the other work that we did is in Drosophila. Drosophila is a beautiful model. It's really almost binary. It's 0, 1. So what we did is we expressed in using different drivers, and I'm just kind of summarizing what we found in the eye, because the eye phenotype was quite striking. If you express K27M with an eye driver, what we saw, whether it was for K27M or for K36M, we saw hypotrophic and very dysmorphic eyes. And actually, even in the context of K27M, there was a baseline of tumors that was created just by the expression of K27M. When we looked at the expression of the marks, as expected, the K27M decrease the di and the trimethylation, and the lysine 36M decrease the tri and the dimethylation of lysine 36. But look what happens to the K27 di and tri in the context of K36M. The loss of the lysine 36 di, exactly as we saw in the mesenchymal stem cell, led to an increase in the di and to some level the trimethylation in those eyes. And the reverse is true. In the context of K27M, we had increase in the K36 dimethylation. What did it do in terms of gene expression? On the transcription level, we saw that a lot of genes that were associated with eye development were downregulated. And we had a lot of genes that were associated with germ cell piRNAs, especially in the context of K27M, that were upregulated. And something that was super striking is that those endogenous retroviruses were humongously, families of them, highly expressed in K27M and even more in the context of K36M. So why is that? If you look at the Drosophila genome, and we did ChIP-seq for K27 tri and K36 di, and we did other marks, but I'll answer a question to that effect. You see that Drosophila has 4 chromosomes. It has a pericentromeric area that hugely enrich in class 1 and 2 transposable elements, and its euchromatin that the chromosomal arm where you have your genes and whatever. And what was very striking to see is that the lysine 36 dye is humongously enriched, and this is true across all 4 chromosomes of Drosophila, in lysine 36 dye, whereas there is barely any K27 tri in this pericentromeric wild type area. What happens when you put K27M? As expected, you have a drastic loss of the K27 trimethylation across the board, except for area that remains like peaky, do not change. But look what happened at the lysine 36 dye. You have a huge redistribution of your lysine 36 dye that leaves the area where it was in the pericentromeric chromatin to your chromosomal arm. It faced area that lost the K27 tri in the chromosomal arm at the expense of those pericentromeric chromatin. And the reverse is the same in the context of K36M. You lose the lysine 36 dye, and you have a similar expansion of the K27 tri, but not to the same level. It does not replace the K36 dye that was present in those pericentromeric arm. And this is what explained the fact that you have a huge expression in K36M of your class 1 and 2 transposable elements, and you have a silencing of developmental genes, because those genes are the one that would specify further eye development and get actually silenced, either by the redistribution or because they are not moving from their initial landing site in the chromosomal arms. And our hypothesis is that this redistribution of chromatin marks the lysine 36 dimethylation taking advantage of loss of K27 di and tri, and the reverse, the K27 trimethylation taking advantage of lysine 36 di loss, is what leads to the aberrant expression of those transposable elements and to the repression of genes that are necessary for specifying eye development. And this is something that maybe we could make work. So just to summarize this part, is in a normal cell, your PRC2 gets recruited to its nucleation sites, start depositing the mono, the di, and tri to coat your epigenome, and works with this boundary, and its boundary being the K36 di, or loops, or other things, and the K27 acetylation, which is super rare. It's really a rare mark that is present there. In the context of K27M, K27 tri do not leave the CPG island shore. It really is restricted there. And the dimethylation leaves as far as the cell cycle time it has will allow it. Or the boundary, if you have a lot more K36 di, it takes more time for PRC2 to deposit the di on lysine 36 di. And this is what will take effect. And this is what we think is leading to two things. This baseline increased ERV expression, especially when you have K27 di loss, and what we think is stalled differentiation, and I'll come to that. So for us, for me, the K27M and EZIP, they just want one thing, is to restrain K27 tri to your CPG island. Everything else is a context of how fast your cell is cycling and what were the boundaries. And maybe this is why there is really very small permissive windows, because not a lot of cells have boundaries that are permissive to the loss of K27 tri and di. So we asked ourselves, okay, are those mutations in the context of high-grade glioma and posterior fossa ependymoma arising in different cells of origin, and between histone 3.1, histone 3.3, in the pons and in the thalamus for histone 3.3, are there different OPCs, or are they really cells that migrated from one area to the other? And this is work that we've been doing for the last few years in collaboration with Michael Taylor's group and Maria, who was a super talented graduate student in Michael's lab, and Celine, who is soon to graduate, a super talented graduate student in Claudia Kleinman's lab. And we've been really trying to chart and do a blueprint, an atlas of the brain development that we did, because at the time we started, there wasn't that much data as there is now. And we acquired areas that were not of interest and times that were not of interest at that time for the Atlas Brain project, and which were the pons, the cerebellum. The cortex has always been an interest, but we acquired also those data from mice and man. And we did single cell transcriptomic, and now we're doing single cell ataxic on these. And we imported every single data set, and there is more than that, that also was published to create our atlas and to create specific signature that would specify progenitors at different time point during development, at different location during development. And this way we could infer trajectories, infer what were the core transcription factor and ways to mature and to go within your lineage commitment. And from there, project transcriptome acquired either at the single cell from the tumor that we study or bulk transcriptomic, and try to infer those signature. And this is exactly what we did for the histone 3.1, histone 3.3 in the pons, and histone 3.3 in the thalamus, and in collaboration with Michael Group on posterior fossa ependymoma. And this is like a huge data set that's going to be available to people, where we try to tease out, okay, is there a different cell of origin? And can we specify what's happening? And what is happening to the chromatin architecture? Because whenever I go to talk, people think that because you have such a huge, what they say, loss in K27 tri, I say non-spread, that they have different chromatin architecture or very bad chromatin architecture. And we do not see that. So the first thing, if you look at the transcriptome data, you see that there is a gradient, that you have anything that's this beautiful orangey and whatever are cycling progenitor that are gradual glial cell, that probably at the origin of what happens. And you see that the posterior fossa ependymoma are more kind of committed in the ependymal lineage. The histone 3.1 really wants to become OPCs, but there is an astrocytic signature that's a little bit more important in here. And that histone 3.3, whether it's in the pons or in the thalamus, are really OPCs with barely any ependymal signature and a little bit of an astrocytic signature. Okay. A lot of things and work actually from Susie's lab initially had shown that those DAPG have higher expression of Hogs genes. And people thought that this is oncogenic. What I'm going to show you is that those Hogs genes are actually a reflection of the cell of origin, the time where those mutations arose. And hopefully you'll believe what I'm going to be presenting now. When we looked, and this work was done in collaboration with Steve Mack and Dipti, a very talented student in his lab, we looked at the level of K27 acetylation and K27 trimethylation, what was pasting in the thalamus versus in the pons, and we saw that DIC and OTX are really highly expressed in the thalamus, and in the pons, Hox genes are really highly expressed. And we look at Hox gene, patterning genes are really, it's the embryo gets patterned, and the timing for this patterning is humongously specific. It really is the clock. It is a differentiation clock and a lineage commitment clock. And beautiful work by the group of Duboul showed that PRC2 is really responsible for this patterning and for maintaining the lineage identity. So you deposit at the transcription factor, K27 tri, you create a specific chromatin architecture that silences forever this transcription factor that's allowing, and this is how your embryo will go and pattern from the anterior to posterior axis with a very specific time point. And what we saw, that this patterning was completely respected in K27M and EZIP tumors. In that, we weren't losing it. Those genes that needed to be silenced because they occurred, it was a Hox gene that, you know, that the mutation occurred probably just after the patterning of this Hox gene. They were here and they were completely maintained. And actually, we can use those Hox genes as a temporospatial zip code to know after when this mutation arose, after when in an OPC lineage did it arise. Architecture is maintained, patterning is completely maintained, and the differential expression of Hox gene is not due to oncogenesis, but just the fact that the mutation arose just after that, and they cannot be patterned anymore. And we could use it to kind of understand when this mutation arose. And this is what gave us the clue for the thalamic tumor. The thalamic tumor do not arise from the pons or elsewhere. They arise in the thalamus proper at the stage of P2, prosomere. I didn't even know about that. We actually imported data that was just published on the developing thalamus. And this is what allows us, those OTX genes that I showed you that were differentially expressed, told us that those mutations actually occurred after the Hox patterning and prior to its being specified at the P2 level. And what about histone 3.1 and histone 3.3 in the pons? And this is where we did the same thing in collaboration with Steve Mack and Céline Jessa. I remember we spent hours just looking at the data, and we came with the answer. There is specific transcription factors that were differentially expressed between the histone 3.1 K27M in the pons and the histone 3.3 K27M in the pons. PAX3 was highly expressed in histone 3.3 K27M, and PAX3 has been identified by Susie's group and by Oren Becker's group. And KX6.1, I didn't even know about it, is something that's really specific to histone 3.1 K27M. And if you want to look at super enhancer, this is a list of different super enhancer that more specify histone 3.1 and those that more specify histone 3.3 in the pons. And this is just to show you using data from single cell epoxy, K27 tri, and K27 acetylation, that those areas that were open in histone 3.1 were completely closed in histone 3.3, and the reverse is true. And actually, NKX6.1 is expressed exclusively in histone 3.1 tumor. This is immunohistochemistry using this transcription factor that's only, if you see it in normal tissues, it's only expressed in the pancreas and barely in some areas in the brain. And it's really enriched and highly expressed in histone 3.1 tumors. And why is that important? Because in your pons, you have the dorsal pons and you have the ventral pons. Dorsal pons is really super rich in OPCs that rely on BMP Wnt signaling. And the ventral pons is super enriched in OPC that rely on Sanji Hedgehog notch signaling and that have NKX6.1 as their transcription factor. And what we think is happening is that those mutations in histone 3.1 are arising in this OPC progenitor that doesn't have access to your oncogenic BMP signaling because it's not soaking in it. And this is why it needs to acquire the ACVR1 mutation that are conferring ligand dependency and oncogenic BMP signaling that's needed for transformation or to maintain stemness. We need to understand better what those ACVR1 mutations, but that's the reason why ACVR1 mutations are superbly enriched in histone 3.1 K27M. And we CRISPRed the ACVR1 mutation in 2 distinct cell lines. And what we saw is that it led to actually a decrease in tumor growth, and it led to a prolonged survival in mice. I wish it was more. I really wish it. I was really disappointed that it's not more, but it's not negligible. It's already a plus. And this goes with some data that were done using inhibitor by the group of Chris Jones and other group like Oren Becker's group. So ACVR1 is needed, but contrary to the K27M mutation, that when you remove it, you really have a drastic decrease, if not abrogation of oncogenesis. Removal of the ACVR1 mutation doesn't seem to be the only way to stop. It gives more time, but doesn't stop completely the oncogenesis of histone 3.1 K27M tumors. What is the function of restraining K27ME3 spread in those cells? I think this is where it's more hypothetical, and this is work in progress, and we have a paper that we're close to submitting on as to why. I think there are 2 states for K27ME3. One is really restricted, and this state can only be achieved in very specific progenitors. This is why we only see those mutations in rare cancers, because not the moment the K27ME3 spreads, and this is the second state, you cannot be oncogenic anymore. And the reason why is normally during normal development, you have a progenitor. It recruits PRC2 to specific nucleation site to allow for patterning and to silence differentiating gene. And you have other areas that are based with the other complexes that I'm not even speaking about today, which are the Swift-SNF complexes, and that are here to kind of modulate chromatin and to express stemness gene. The progenitor receives cues to differentiate or to further lineage commit. Your PRC2 is nimble. What's patterned remains patterned, but other area that needs differentiation can get access to differentiation, especially your PRC2 can go and silence stemness gene. And this is what's not happening in those tumors. Within the given cell cycle, your PRC2 is unable to do its function and just is able to deposit the K27 tri there and is not nimble anymore. And this loss of nimbleness is potentially what is causing those tumors from arising. And you have such a tight gene repression at those areas where K27 tri is restricted that you cannot hope for further differentiation or further lineage commitment. And if you needed further convincing, and this is something that was published also in our paper, Brian Krug in my lab, when he CRISPRed those mutations in differentiation media, even if when you have K27M, you can barely express GFAP. Look what happened when you remove it. You already have GFAP at baseline. And when you push the cells to differentiate, they differentiate and they don't create tumors. And actually, in the brain of mice that were injected with a knockout to the K27M knockout line that weren't dying of tumor, those mice were alive 1 year after the injection. We can find human DNA, and we're trying to see now into what they were differentiated. And this holds true now for 4 distinct cell lines, multiple clone within the cell lines. So this is something that I'm quite comfortable presenting. In the last 5 minutes, I'll go very fast. It's unfortunate because G34R, V, and W are fascinating. They really are a kind of another way of mediating oncogenesis that we're only scratching the surface. And I'm going to try and show you very rapidly that a lot of work is being done and a lot of cues are being there. As you see, the W never arise in the brain, and the R and V mainly arise in the cortex, in the brain cortex. And they are exclusive to histone 3.3. So one first cue, and a lot of people have done that, is that probably the lysine 36 downstream of the glia lysine 34 is not being properly post-translationally modified or read. And one of the work by Peter Lewis, and we collaborated in that, showed that the NSD2, which deposits the lysine 36 dimethylation, was inhibited by any of those mutations. And the fact that you had less lysine 36 di being put, remember what I told you, the di is a boundary for K27 tri, you had more PRC2 going there and you had undue silencing in that. Okay, and I'm gonna go fast, this is published. When we looked at the histone 3.3 G34R gliomas, we had a beautiful surprise, and this work was published in Cell 2 years ago, a year and a half ago now, and was co-led by Celine, Jihad, Shreya, and Carol in my lab. And we showed that PGFR-alpha mutation and overexpression was preponderant in those tumors. 50% at baseline, 80% at recurrence had PGFR-alpha mutation, and the expression is throughout your cohort. And Susie had shown that those PGFR-alpha mutations that are identified in high-grade glioma are really in the extracellular domain of the kinase, and this is something else that we need to understand. And indeed, those mutations were really hugely enriched there, and you had barely any mutation in the kinase domain. And just to give you an idea, barely 6 to 8% of the other glioma have PGFR-alpha mutation, and more than 50%, and I'm telling you 80% at relapse of the G34 mutant have it. And what we showed is that those were not gliomas. Those tumors were arising actually in interneuron progenitors, either prenatally or just postnatally in the ganglionic eminences. And when we tried to the interneuron rise from the ventral forebrain in the lateral ganglionic eminences. And the moment you have those transcription factors that specify interneuron progenitor, you really suppress any oligodendroglial differentiation. This is why olig 1 and 2 are never expressed in those tumors. We have the answer to that. But to reconcile how come PGFR alpha, that is actually one of the major oligodendroglial kinase, receptor tyrosine kinases, was apparently expressed there, we had to go to chromatin architecture. And what we showed is that actually those tumors were co-opting something that was present during development. And there is actually a loop that's formed between the PGFR alpha promoter, GSX2, which is the main transcription factor that specifies interneuron progenitor identity, and the enhancer of this GSX2 that's present in development, but usually silenced at the level of PGFR alpha. And only GSX2 gets expressed in ganglionic eminences. This loop is being taken advantage of, and by some reason, and we need to understand why, PGF alpha gets expressed. And this is how we have oncogenic tumor formation. And I'll end by one thing. We are modeling the G34 mutation, and if you need to be convinced that a residue could be different from another, we did it on two distinct genetic backgrounds. The G34 R, V, and W mice has distinct phenotype. R has a neurodegeneration, W nothing. And W has obesity and bladder dysfunction. And this is something that we're further teasing out, and we've done a lot of things. I'll go fast on this because I think this is just to show you that once you have the good question and the right tools, you can work. And there are actually germline individuals, germline mutation in G34 R and V that have microcephaly, neurodevelopmental delay. And this is something that we're further kind of going into, and those are the individuals that are there. And I'll end up, I need to say that this work is through the amazing people in my lab, and in Claudia's lab, and in Jasek's lab, the amazing collaborator across the world, Steve Mack and his group, and other people that collaborated and contributed. And of course, our funding sources, the NIH is funding our mouse work, the Genome Canada is funding our single cell work, and we're recruiting if anybody's interested in what we have to say. Thank you.
Fantastic, Nada. If people have questions for Dr. Jubato, just type them into the all panelists, and we'll ask them. And she's happy to stay past the top of the hour to answer any questions that there are. Let me start with one, Nada. You mentioned, I think, K27 acetyl role in viral mimicry and in repression of the transposable elements, but said that it differed, I thought you said, in the EZIP versus the other models. Did I hear that right? And then how do you think of a mechanistic contribution if it's varying across the models?
So I think the viral mimicry, or I would say the pervasive acetylation occurs only when you lose a K27 dye. And the K27 dye, you lose it when your cells cycle faster. And this is in high-grade glioma that are histone 3.1 or histone 3.3, and maybe EZIP positive. But the posterior fossa ependymoma probably cycle, even if they are very kind of aggressive, they cycle slower. And you have time to deposit the dye, and really it's coated throughout. And this is why we don't see it. Even in the histone 3.1 K27M mutant, the dye methylation was really across the board. So it is actually a function of how much K27 dye you're losing in the cell.
Got it.
Not a function of the mutation.
Got it. So Dr. Carol Thiel, hello, Carol, says, when you speak of boundaries, are these defined by higher order chromatin structure or rather by the pattern that you see, air quotes, normally?
So we've done extensive analysis of the high C, and if I can tell you that we have 30 tumors, lines, and whatever, if not more, there is minute changes. It really, if you compare it to other progenitor, there is really no changes in that. So the loss of K27 tri is not affecting the chromatin architecture. There's something. It's a little bit more subtle, and I think it has to do, but it's not major changes in boundaries and whatever. We need to do better due diligence for CTCF, But my thinking is that you're not kind of moving a lot of the CTCF and whatever, and we won't have surprises there. But this is something that we need to do better due diligence about.
Great. Dr. Susie Baker asks, it's very interesting that the G34R and G34V mutations have different phenotypes. What's the mechanistic basis of this? Is it related to the effects on 3D chromatin architecture that you highlighted for the G34R mutation?
No, it's not at all. So we have like a beautiful analysis on this. Again, this is a work that's in revision. I hope it goes. I'll share it. It's not like there are no secret. They have differential effect on the lysine 36 downstream. And it's actually, this is when you overexpress, you lose, there's not enough, it's true up to a point, but you have it, when you have it in dosage and it's same, this is where you see the effect on chromatin. And actually, they really impact differentially the lysine 36 dye that's downstream. And potentially the lysine 27 and other mark, because when you impact one, as you saw, it's a domino effect one way or the other. And we have like strong clues as what's happening there as to why.
Great. Next question. Can you speculate a bit more about the developmental window? You mentioned that it may be chromatin landscape. What are you thinking about genes versus cells?
Okay, so what I think about, and again, I tell myself story. This is maybe why I am a pediatrician, because I like to kind of tell myself stories. Chromatin, people say epigenome reversible and whatever, it's true up to a point. And if you have the endless time, this is why we needed actually to put HTERT and other things in order to kind of prolong the time of a given cell in order to do the reprogramming. And we still are struggling with reprogramming because it's very ineffective, which tells me that this plasticity is true up to a point, especially when you went into further differentiation. I think that once you have your K27, and this is only me, and please don't, you could quote me, but I don't have yet enough proof to kind of show it, but this is what I think, is that K27 trimethylation mark is a mark that's very hard to kind of back up. The lysine 36 dye, when it recedes, it's not very easy to bring it and have it back. And even when you have a mutant that's hyperactive that deposits the lysine 36 dye and pushes, it is very hard to have K27 try change its mind and change its location. So I think this is what I mean by specific developmental window. The moment your K27 try spreads, your likelihood of becoming a cancer decreases drastically.
Wow. Great. It's just been so fascinating to see the mechanistic insight come from this. I do want to mention we're at the top of the hour, so this is the formal end, but everyone's welcome to stay on, and Dr. Giabato will continue to take questions. If anybody wants to add questions in, just send them to all panelists. The expression of Hox genes in terms of identifying which rhombomer from which tumors arise, I think of work by Andrew Lumsden, Rob Krumloff, Denis Duboul. So is this like VDJ recombination in as much giving us really the zip code that you're talking about? Are you confident the Hox codes are stable and actually telling us all the way back to the cell of origin?
I think they are stable up to a point. I think they don't tell me when it happened. They tell me after what patterning it happened. Once you get your patterning, you're not losing it. This is the message that I'm saying, is that you're not losing patterning. The loss of K27 try do not occur on things that have already been patterned. Okay, I don't know after that when it happened, but this is, and probably this is the reason why we see it in adults. I could know it happened after this patterning, and then when does this beautiful K27M find its partners, because it needs partners in crime. Maybe the reason as to the time, the delay, and we have so much more to learn from that. But I'm quite comfortable with the different transcription factor, with the different OPC lineages. The work that we've done, it's not everybody does a beautiful work, but we really took our time and we did gazillion permutation, multiple different tumors, not only lines, but tumors, just to kind of be convinced that this was it.
Really interesting. Again, next question. Is it possible to determine the mechanism of cancer driving transcriptional changes? How much hypersilencing contributes versus failed silencing versus both?
I don't know. I don't know. I wish I knew. What I think is that the K27M, and again, because I only focus on K27M and EZIP at this point, what you want, remember what I told you about K27, try when it's unleashed, it's hard to kind of recover from that. If you want an embryo or a given tissue, whatever, to have the number of cells, the number of differentiation, and the tempo of differentiation, you need to have checkpoints to stop the K27 try from spreading or unduly spreading. So this is why those mutations mimic something that exists that we need to understand a little bit more. This is why the K36 die boundary is so important, because it really also space how fast your cell can spread. So I think silencing of specific gene is quite key. That was a question. Is it silencing or expression? But then the counterpart of this, if you silence something and prevent something that can silence other things from moving, you get undue expression or too much of prolonged expression of something else. So this is my very naive way of saying it. It's a compound way.
Yeah, it's interesting, along those lines, interesting that, it can be both gain of function mutations in EZH2 and loss of function mutations in EZH2 in cancer. So the context dependency is important, but I think you're dissecting it beautifully here. Next question. Can you distinguish between mutations only occurring in spatiotemporal regions versus they can occur anywhere, but only drive cancer when they happen in one zip code?
I don't know about that. I really don't know. So this is something, so we have, and I think Susie also has a disinducible model of K27M. And we're trying to kind of create one for EZIP, but it's going to take a lot of time. And I don't know how this is something that I'm struggling with. It's why, so we're trying to kind of put them in different organ. Why am I not seeing it in, let's say, in lung cancer, prostate cancer, in other sarcomas and other, I don't know. There are some rare cases of osteosarcoma that have K27M and that have G34W, but we don't see it elsewhere. So why it's also, I'm kind of hiding behind what I say, chromatin architecture, permissive window. We really need to kind of define it way better. So this is more work to be done. I wish I had like Charles Mulligan's, the amount of leukemic cells and whatever, and the different ways to kind of, he's doing beautiful work to that effect. And this is something that I'm quite envious of at multiple levels.
Agree, it's fantastic work, and I think he's here listening and fascinated by your work as well. Others, feel free to type in any questions. Just one other one was a question about the pericentromeric K36 dimethyl changes in the K27M mutants, and thoughts on mechanistic contributions to driving cancer there.
So I don't know. So again, the Drosophila is beautiful in that it doesn't have DNA methylation. DNA methylation and lysine 36 dye, one of the things that was shown, and this is work we kind of collaborated in, it's mainly the work of Chao Lu, showed that the lysine 36 dye recruits the NMT3A. And in the intergenome, this is how you put the de novo methylation that transform into maintenance methylation. Okay. We don't have this layer in Drosophila. And for me, it was very exciting to see that just by removing the lysine 36 dye, we had this huge expression of those retroviral endogenous, those transposable elements, like huge. Are they contributing to oncogenesis? I don't know. Do they have new integration? We're looking into that. This is the K36M lines are a nightmare. You don't have them. This mutation is badly tolerated, probably because it creates such a genomic instability that it's impossible to tolerate for a cell. Still, you have cancer that have it. So we're trying to kind of decipher all of that as we go.
Great. Next question comes from Dr. Mulligan, saying, Nada, we saw rare histone mutations in TALL.
Yes, I know.
Sort of seeing a phenocopying of the EZIP raises the possibility that it may have a role in leukemia also. How high might expression need to be oncogenic?
You don't need it to be very high. You really don't need it to be very high. I think 5% of your total histone 3 pool is more than enough. And this is something. So again, I saw those two, and they had the IL-7 receptor alpha, if I remember, as their added oncogene for one of them, and the other one I'm blanking. I looked at that quite in depth. I tried to see if there were more, but there were none.
Great. There are a number of comments of people saying how much they enjoyed your talk, but we've come to the end of the questions, and so I just want to thank you, Nada, for a really fantastic talk. Great science, provocative things that keep all of us, even if brain tumor and elsewhere, interested.
Is Susie still on the call?
I can't directly see whether Susie is still on the call or not. She's won't be unfortunately we don't have her as a panelist, so she won't be able to unmute.
It's okay, because I felt bad about not addressing completely the question that she had, but it's fine. I'll speak with her at the
She's not still on. She must have had a 1 o'clock. Sorry, there's one more that just came in. Let me catch it. It just came in. Fantastic talk. My understanding that the proliferation and differentiation of progenitor cells is precisely coordinated during critical developmental windows. When proliferation and differentiation programs become uncoupled, degeneration, dysplasia, or cancer can occur. The epigenetic changes may play a more important role in activating differentiation genes than in silencing progenitor or proliferation genes. So is it possible that the G34R, G34V, or G34W, I think, causes differential epigenetic changes? I think you were beginning to allude to that.
Oh, they do. Oh, they do. They really do. And again, because this mutation really acts in cis. So the K27M, even if it's not incorporated, it works and it prevents EZH2 from functioning correctly. The histone 3.3 G34 needs to be put in cis, meaning incorporating in the Chromatin. And it's funny, nobody asked me the question, why histone 3.3 exclusively for G34? You have your answer in that histone 3.3, whenever you want to do something in your genome, either express a gene or repress an endogenous retrovirus or do something more active than just kind of filling and kind of compacting your chromatin, you put the histone 3.3. And because it's actin cis, this is where the damage comes. And as you further go and you further differentiate, your cell cycle is not the most predominant thing, exactly as this person was alluding to. And you have way more histone 3.3 being deposited and way more effect. And I think this is how we see the effect of the G34 mutation in the interneuron progenitors. It needs its time for them to just stop differentiating, and then it will matter, and it almost like a small alien, it will go and do its wrong work.
Great. Hong-Lin, thank you for that question. All right, now we are at the end, Nada. So thank you again. Fantastic talk. And
thank you for inviting me.
Soon.
Thank you very much.
Bye.
Bye.